Welcome everyone to session 18 STEM of the 7th Polygen Symposium of Rising Scholars. I'm going to be the moderator today, Veronica Edwards, and I'm an employee at Polygence, currently working in data analytics, and super excited to see your guys' projects and hear you guys present. The goal of this is obviously to celebrate and showcase like all of you guys' hard work over the past few months. And I know that like you guys have put a lot of effort into your projects, and I'm just going to be able to see a glimpse of that today, but I'm really excited to hear. And so we just have you two for now. Hopefully Maya will join us in a bit, but if not, then I'm sure we'll have two great projects to listen to. A fellow Polygence member will be scoring each of your presentations today, which will help determine like prize winners after the event. So stay tuned for that. And yeah, if any audience members have questions during the presentation, please type them into the Q&A. So yeah, we're going to get started, and we'll start with Serena. So if Ayush, you could mute and stop sharing your, or not stop sharing, pause your video. I will spotlight you, Serena, and go ahead and share your screen.
All right. Okay, can you see my screen?
Yes, I can.
Okay. Hi, everyone. My name is Serena, and my project is called Techniques to Induce Cardiac Regeneration Post-Myocardial Infarction. All right. MI, colloquially known as a heart attack, is caused by a major acute decrease in blood flow to the heart. And this can be caused by a number of reasons. Most commonly, it's a blood clot or a buildup of plaque in one of the major arteries leading to the heart. MI is one of the leading causes of death in the United States and worldwide because it's not just an acute problem. MI leads to major necrosis or cell death in large parts of the heart. So the heart can no longer function efficiently, which leads to long-term and life-threatening side effects and a very poor prognosis. It's almost impossible to pinpoint the cause of a heart attack, but there are many lifestyle and genetic risk factors. The treatments that currently exist for MI are usually inefficient or invasive and meant to prevent future MI instead of treating the problem that already exists. So on the right, there's a picture with a few common interventions for MI, and these can all actually be minimally invasive, but they're not long-term solutions. So individuals receiving these treatments are still at a much higher risk of MI for the rest of their lives, with an increased risk of death for at least 7 years post-MI. Cardiac cells are notorious for their particularly low turnover rate, meaning they don't duplicate and replenish themselves as fast as the rest of the cells in our body. And so the heart never fully heals itself after a cardiac event. In recent years, cardiac regeneration has been a hot topic of research to address this problem of poor natural regeneration. One of the techniques on the forefront of this research is gene therapy, specifically with the use of CRISPR-Cas systems. So the CRISPR-Cas system is found in the immune system of many bacteria and archaea and is used for their natural viral defense. If we take a quick look at this diagram, we can see a strand of DNA, the Cas9 protein, and a guide RNA. The guide RNA directs the Cas protein to a specific point that it's been programmed to detect, and the Cas9 protein cleaves the DNA at that point, rendering that gene inactive and unable to harm the host organism. This process has recently been adapted for use in humans. So how can we put it to use in regenerating the heart? One way is to reprogram cardiac fibroblasts into cardiomyocytes. So these are the two main cell types that make up the human heart. Cardiomyocytes are responsible for heart muscle contractions, which keeps the heart pumping and the blood flowing. Yet they only make up about 30 to 40% of the cell population in the heart. Fibroblasts, which make up the remaining 60 to 70%, are responsible for the basic structure and electrical signaling in the heart. Post-MI, the loss of cardiomyocytes affects the heart's ability to contract and therefore pump blood. A 2010 study found that in vivo introduction of a combination of transcription factors that are crucial to cell development, namely GATA4, MEF2C, and TBX5, could be directly reprogrammed into cardiomyocyte-like cells that have a very similar structure to cardiomyocytes. And here we have a basic diagram of what that would look like. Another intriguing technique to regenerate the heart is through the use of induced pluripotent stem cells. Stem cells are the body's basic cells that differentiate and specialize into their unique cell types as they grow. They also have the ability to self-renew, unlike cardiomyocytes. In 2006, Yamanaka and Takahashi found that with the addition of certain transcription factors called the Yamanaka factors, somatic cells or already differentiated body cells could be turned back into stem cells, and they would be called induced pluripotent stem cells. So these cells can then redifferentiate into a number of different kinds of body cells. The use of stem cells in cardiac regeneration presents the possibility of differentiating stem cells into cardiomyocytes, which can then be transplanted into damaged post-MI hearts to promote regeneration. So I'm going to talk about a few studies. In 2009, we et al. Found that with the embryoid body method of differentiation, cardiomyocytes can be created. So in this method, spherical structures are created that are very similar to human embryos, containing all three germ layers. In 2018, Go et al. Showed that bone marrow mesenchymal stem cells, which are multipotent and can differentiate into a few specific cell types, could be induced into pluripotency to then create cardiomyocytes. Lastly, a 2022 study by Schwartz et al. Examined the effectiveness of two synthetic modified mRNAs in promoting cardiomyocyte regeneration, and these were Stemin and the mutant YAP5SA. In the study, post-MI mice models were injected with these two modified mRNAs, and 1 month post-MI, the mice heart showed expression of a number of stem cell factors that allowed the senescent cardiomyocytes to be dedifferentiated and able to replicate again. While most of this research and these techniques seem very promising, there are still some limitations that must be addressed before widespread use and a move to clinical applications. So first off, CRISPR has a large capacity for off-target effects. There's always a possibility of unintended genetic modifications when knocking out or replacing genes, and this could affect the genome. There's also some uncertainty about the best delivery mechanisms for CRISPR into the heart. Stem cells also have their fair share of complications when being used in the human heart. First off, there's a lot of debate within the scientific community about the ethical uses of stem cells, especially embryonic stem cells, which are pluripotent and often used in research, and they come from the blastocyst phase of embryos. There's also an extremely low efficiency with inducing pluripotency and differentiation, showing less than a 1% efficiency in early studies with not much progress since. And lastly, the inflammatory environment of the heart does not provide a suitable place for stem cells to properly renew and repair the heart. There's currently some ongoing research about these techniques in clinical trials. One study that aims to be complete in 2023 is studying the use of human-induced pluripotent stem cell-derived cardiomyocytes to improve left ventricular systolic function, or LVEF, in patients with ischemic cardiomyopathy. So in the study, patients receive allogenic transplants of induced pluripotent stem cell-derived cardiomyocytes, and are assessed for a number of measures, including improved LVEF, adverse effects, and abnormal vital signs. A clinical study that was completed in 2015 tested the safety and efficacy of adenovirus-delivered VEGF-D gene therapy in improving the myocardial perfusion in patients with severe coronary artery disease. It was found that the treatment was safe, feasible, and showed a statistically significant increase in myocardial blood flow. The use of CRISPR-Cas systems and stem cells have provided major breakthroughs in disease modeling and regenerative medicine in recent years. And while there's still a long way to go before these techniques are approved for clinical applications, at the current trajectory of research, their use will become increasingly widespread in the coming years. Thank you.
Awesome. Thank you so much. That was incredible. I'm
Thank you.
really glad to have listened to it. And I think, let me see if I can unspotlight you, but we can ask some questions first. Yeah, I'm curious, how did you get into this research topic? Like what drew you to this subject matter?
Yeah, so I've always been interested in regenerative medicine. I just think it's really cool and futuristic. But last year, my dad actually had a heart attack, and he's fine now, but it combined, I was able to research more about regenerative medicine and specifically for something that affects him and my family. So it was really satisfying to complete this project.
Yeah. And were you able to, oh, sorry. That was my alarm to tell you you had a minute left. Yeah, were you able to kind of, use your experiences, I guess, and that influenced what you would look into in this project?
Yeah, definitely. My experiences definitely influence what I would do with the project. I knew I wanted to do something with CRISPR and stem cells, but this was the perfect way to combine that.
Awesome. And what would you say would be, I guess, an unexpected part of your research, or something that you found the most interesting?
Probably, I didn't know the extent to what these techniques were already being used for. There's a number of clinical trials where CRISPR and gene editing and stem cells have been used and successfully treated certain diseases. And obviously they're still in clinical trials, but I didn't realize they were actually they were up there. They're almost there.
Yeah, you're definitely doing research at a time where there is a high volume of people doing these types of research, and there's discoveries happening all the time. So definitely an exciting time to enter this field.
Yeah.
I guess I'll ask you just one more question before we go to Ayush, and then we can ask a few more general ones. But do you know what, if you were to continue doing research into this field, what would you study? I know you mentioned future directions for this types of research, but do you want to continue studying more about this?
Yeah, for sure. I would love to continue studying more about it. I would love to continue studying about it in more a clinical setting and actually see CRISPR and stem cells being used in action for sure. Cool.
I would love for you to continue as well. And it's great that we have people like you interested in this in the world, so that we can make progress. So that's great. Thank you. Thank you so much for your presentation. It was great listening. Awesome. So next we have Ayush. I think, can you turn on your video? I don't know if, okay. Oh, perfect. Hello. Great. So you have around 10 minutes, and feel free to go for it. I will make you the spotlight for everyone. I cannot hear you.
How about now?
Yep, perfect.
Okay, and you guys can see my screen?
Yep.
Great, okay. My project is called Landmine Locator. It's a potential autonomous landmine detection algorithm using computer vision. Active landmines that are left as remnants of war have been plaguing countries for decades, and annually 5,000 people die per year at the hand of landmines, and 30,000 Afghanis have been killed by landmines since 1978, with children being a vast majority of the victims. Right now, the current methods of solving this problem use expensive ground-penetrating radar-equipped vehicles that are just simply not designed for civilian use. With the use of a cost-effective civilian-operated landmine solution such as mine, people can clear out that no destruction will occur. Use of my drone will be able to map with an identifying landmine algorithm using the camera and field of view. My engineering goal is to test the feasibility of building such an autonomous drone and analyze its performance for detecting landmines in war zones. The drone's performance will be determined by accuracy, perception, planning, and action. And to do this, I will demonstrate that you can plan a path for the robot given some constraints and execute that path while updating where you should be along the path based on critical information like battery health. What is the problem and why is it hard to solve? Well, in the 80s, over 1 million PFM-1 landmines were dropped into Afghanistan during the Soviet-Afghan War. These landmines have green wings that allow them to flutter to the ground, easily detonate, and are mostly made of green plastic, making them both attractive to children and difficult to find with traditional landmine detectors. More than 30,000 Afghanis have been killed or injured by these and other landmines and unexploded weapons since 1978. And in recent years, children have accounted for the majority of these victims. This problem is particularly hard to solve because there are so many landmines around the world that are very neatly hidden so that people and animals just don't see them. Not only that, but the current methods for landmine removal typically require people to trigger the landmine to know that they're in the area, and then people use other devices to go out and search for them. The countries that have the most landmines are also the countries that don't have the money to support a program to get rid of them. And because of that, the death rates soar annually each year. Landmines are inhumane because by design, they inflict brutal damage to the human body that can kill or create lifelong injuries. And once planted, landmines don't go away until they're removed. Landmines deployed during World War I are still causing death today in Europe and North Africa. There are a couple existing mine detecting and detonating drones, such as the Mistral Hound, but it's only sold to the US Army and Special Forces. A company named Mine has developed 2 drones that are capable of mapping, detecting, and remotely detonating the bomb. And their products are available for civilian purchase. However, these products are particularly expensive, and these companies prefer organizations or large companies buying their products. My solution is to build a drone capable of identifying landmines. The drone flies into the potentially contaminated area, scouts the ground for a potential landmine, and if identified, the drone will take a picture of the mine and send back the GPS coordinates. The user inputs the length of the field that the potential landmine is in. The drone then flies right to left along the x-axis of the field until it reaches a corner, to which it then moves up the field by an amount inputted by the user. And then it flies right to left, and this process is repeated until the drone reaches the end of the field on the left side. And with the flight path being this way, the drone can scout out the most ground, increasing the chances of identifying a landmine. This is a picture of my original function structure diagram. And in this, there are 3 overarching modules: the state, goal, and the controller. The state module is responsible for constantly checking the state of the drone, and things that it checks for are the altitude of the drone, the airspeed of the drone, the speed of the propellers, the battery life, and the current GPS coordinates. And next we have the goal module. And the goal module takes in the information from the state module and basically says, so this is what's going on with the drone. So with these parameters in mind, how do I get where I want to be? And it figures out what movements are required for the drone to continue on to the lawnmower path. And next we have the controller module. So the controller module is basically responsible for executing the commands on where to move that is given by the goal module. And the controller module is what physically moves the drone. And this is a much more refined function structure diagram, which has all the smaller functions that come out of the 3 overarching modules: the state, goal, and controller. We have things like the speed, the camera, the heading angle that's fed into the controller, and the time. This is a screenshot of what's outputted when I run my lawnmower pattern algorithm. As you can see here, the user inputs the bounds, the X, Y, and Z bounds, and I inputted 50. It then asks for an emergency stop percentage, to which I entered 15. And what that basically does is that's a fail-safe that I implemented. So if the drone ever reaches that battery percentage of 15, it just won't take off, because then it won't have enough battery to be able to return back to where it took off. So the computer returns all of that, and then it does some math, and it basically calculates with the X, Y, and Z bounds being 50, the total length of the pattern is going to be 1,300 units. And then you can also see here the coordinates that is outputted. Basically, each one of these coordinates is where the drone has to move, and once it moves one after another, it eventually completes the entire pattern. Software and technology that I use. I used the Parrot Sphinx simulator for all my testing. This is a simulator that was provided by Parrot themselves. To program within the simulator, I used the Parrot Olympe SDK. That was also a SDK provided by Parrot. In order to get access to both of these programs, I had to buy the Parrot and Nafi drone. And to run these programs, I used a custom-built computer with an RX 580 graphics card. I used Linux 20.04, and then within Linux, I used a VIM text editing program, and I coded in Python. And I also used proprietary libraries that I had to receive special access from Parrot themselves. And I basically sent them an email, and I explained them my project, and I gained access to that. Some constraints that I had and plans for future development. Constraints, the Linux installation proved to be very difficult and long. I had a lot of installation issues because I basically installed Windows and Linux at the same time, and that proved to be very difficult. I also had a lot of Wi-Fi problems after Linux installed. The Wi-Fi chip in my PC proved to be pretty much useless because there was always a driver error when I moved from Windows to Linux. So I had to use an Ethernet cord the whole time, which proved to be very annoying. And I was also unable to connect to the virtual drone for about a couple of days. I had to work with my mentor to be able to fix that. And just like anyone programming, there's a lot of errors in my code initially. So I had to spend hours constantly debugging that, but eventually worked out. And then both of the programs, Olympe and Sphinx, are extremely demanding. And I learned that when I tried to install those programs on a lot more or less powerful computer, and it just simply crashed the computer. So I was always forced to use my my desktop PC. And plans for future development, I want to be able to use computer vision to be able to detect different objects and not just small landmines. And I eventually want to get out of the simulator phase and run the program on my physical drone, the Parrot and Nafi. And that's all I got. Thank you.
Wow. Thank you so much. That was so interesting. It goes to show that people or polygents really have a variety of interests. So thank you so much. I have a few questions from our audience for you. So let me see if I can get that up. So do you envision this being used by civilians, military, et cetera? Like who would be the ideal audience for your project?
Well, the ideal audience would be probably civilians in an area where there's a lot of landmines. So like in my project, I mentioned Afghanis. It'd be really cool to be able to see the people themselves actually use my project. And I think this could definitely help them a lot.
Awesome. Yeah, that makes sense. And I feel like it could be, used worldwide and all around the world. Like you mentioned, there's so many different issues in different countries and whatnot. So that's great. And I know you mentioned at the end the future developments. This is another question we had. Do you know what you think like, what do you see as being the next big question to address like in this field?
I'm not too sure what you mean by that question.
Yeah, I guess the way that I interpret is like how would you what would be the next thing that you would have to answer to continue doing research in this field? Like, are there any barriers of entry? Are there anything like you mentioned right now you're in the simulation? Like, how would you get to being able to actually conduct this type of research with a live drone?
Yeah, so the first step that I'd have to do is probably getting some form of like actual landmine that I can test with. So I'd probably make some kind of explosive myself and probably have to test with that. And there's a lot of constraints with that, such as like safety, obviously, and I need to find a spot that I can actually detonate a landmine. And there's also a lot of safety aspects with the code and the drone. If something goes wrong with the code, then the drone could start doing random things and just fly indefinitely, and that would obviously be really bad. So yeah.
Yeah, you mentioned you also had to email someone to get access to certain. Oh, are you still there?
Oh, I think my camera went out.
Oh, no worries. I'm just wondering if, you had to get access to a certain type of programming to even be able to do this project. Like, has it been difficult in that regard as well?
Yeah, in order for me to get access to the Sphinx program and use the libraries, I actually had to send an email to Parrot themselves explaining my program and what I hope to achieve. And that was a little difficult. They didn't really take me seriously at first because I'm just a high schooler, but I eventually explained it to them and they sent me all the stuff I needed. So it eventually worked out.
Awesome. I'm glad they let you have access to it. And I mean, it's great that it's so incredible that a high school student could, get access to that and use it for something meaningful. So great job. I loved hearing your presentation.
Thank you.
Great. So it looks like Maya is here, which is awesome. Maya, can you hear me?
Yes, I can.
Hi.
Sorry
Okay,
about
great.
the technical difficulties.
No worries at all. We are very happy to have you here. I'm glad you made it in.
Yeah.
And it's perfect timing as well. So you are the third presenter and we have time for you. So gather your thoughts. Do you have a tab open with your presentation?
Yes.
Okay,
I do.
great.
I'm just on a MacBook. I figured it out, but I'm not nearly as used to this sort of setup. So I'll
No worries at all. This is a confusing day. There's been a lot of technical difficulties, so it's not just you. But yeah, if you are ready, I will put you on a spotlight and you can share your screen and you'll have around 10 minutes. I'll let you know if you run over, but yeah, and then we'll have some questions from the audience for you and we'll go from there. Sounds good. Okay, cool. So I will spotlight you and
let's see, I'm gonna share my screen. Okay. Cool. Oh. Sorry.
No worries. You can unshare if you want to redo it.
Oh, sorry. There we go. Okay. So, hi, my name is Maya, and this is my presentation on the etiology of schizophrenia, as well as the role that genetics and GRIN2A play in it. So, schizophrenia is classified as a psychotic disorder, typically involving changes in a person's thoughts, feelings, and behaviors. According to the DSM-5, there are several different types of schizophrenia, including paranoid, disorganized, catatonic, residual, and undifferentiated schizophrenia. And some of the differentiating factors between these types include whether or not a patient experiences hallucinations, which is common with paranoid and undifferentiated schizophrenia, and whether or not a patient experiences difficulty in, say, motor control, which is common with disorganized and catatonic schizophrenia. And then predicted causes of schizophrenia include drug use, trauma, stress, and genetics. But I do need to address one thing, and that is there's a lot about this that we simply don't know. Either we don't have the technology and tools to research it, or the research simply hasn't been done yet. And so we are doing the best that we can with the information that we have, but it can be pretty limiting. So schizophrenia can have positive and or negative symptoms, and it sometimes depends on the type of schizophrenia within a patient. For example, positive symptoms, meaning that they're sort of adding on to a person, can include things such as delusions and hallucinations. However, negative symptoms, meaning that they're sort of taking away from a person, can include things like diminished emotional expression and diminished motor control. And these things can tie to the type of schizophrenia. For example, paranoid schizophrenia typically has more positive symptoms, and catatonic schizophrenia typically has more negative symptoms. So you might be wondering what this has to do with GRIN2A and genetics, and what exactly am I getting at? Well, I will say again that there's a lot about schizophrenia that scientists don't know, period. But every day we do get closer and closer to figuring out the causes of the disorder, including genetics. A 2019 study done by researchers at the University of Helsinki, as well as the Broad Institute of MIT and Harvard, uncovered consistent mutations across over 10 separate genes, all connecting back to schizophrenia. One gene of particular interest was GRIN2A, which interestingly enough is related to a few other disorders, including attention deficit hyperactivity disorder and epilepsy. So GRIN2A is in charge of encoding for an N-methyl-D aspartate receptor subunit targeted by the excitatory neurotransmitter glutamate. Glutamate has in fact been suspected to be related to schizophrenia in some sort of way, as several drugs that block glutamate receptors, such as cannabis and LSD, are known to have symptoms similar to those of paranoid schizophrenia. Not only that, but because NMDA receptors are integral to the neurotransmission of excitatory inputs, a dysfunction in them could lead to an imbalance of neural excitation and inhibition. Such factors have an established connection with neurological disorders such as schizophrenia. And then my graphics here, on the left is the structure of glutamate, and then on the right is a depiction of the transmission of glutamate. And right here in the middle is what GRIN2A does. It encodes for a subunit of the receptor NMDA or NMDA-R NMDA receptors. So there is some progress being made in terms of targeting GRIN2A, yet all are primarily focused on its connection with epilepsy. For example, the Cure GRIN Foundation uses tools such as AAV vectors, which are adeno-associated virus vectors, and CRISPR, which stands for clustered, regularly interspaced short palindromic repeats, thank God for the acronym, in the form of gene therapy. And then researchers have recently attempted to target genes that encode for NMDA receptor subunits, such as GRIN2A, by drug screening on human neurons. Such drugs include L-Serin, as tested by a 2020 Barcelona study. What this could mean is that by knowing what genes are related to the development of schizophrenia, researchers can thus begin testing gene therapy. Tools such as CRISPR and AAV vectors can help reverse phenotype in target cells. This is huge news, as gene therapy early on could be a definite edge in mitigating risk. Also, since GRIN2A encodes for a receptor subunit targeted by glutamate, scientists have a lead in researching that specific neurotransmitter and its relationship to schizophrenia. This basically means that we're getting somewhere. And then the point of this presentation was to partially provide information about a disorder that most people won't admit they don't know a lot about, but also it's to sort of inform others about the specifics of this disorder. So schizophrenia has over 5 different types, and there are several key symptoms that include hallucinations and paranoia. But in this presentation, I went deeper to talk about the role that genetics and one gene, GRIN2A, plays in schizophrenia. GRIN2A has a connection with neural excitation and inhibition, as well as glutamate, all of which have established ties with schizophrenia. Ultimately, this is a big step forward in truly understanding the disorder that is schizophrenia. Thank you.
Thank you. That was great. Thank you so much for sharing. I was wondering if you could share a little bit about how you got interested in this topic.
Yeah. So schizophrenia does run in my family. My grandmother had it and my uncle has it. And so that sort of got me interested in how exactly it's passed down. And I mean, is there some sort of gene that is related to schizophrenia? And if so, is it possible that I could have gotten it when I was born? And all of that sort of snowballed into me just deep diving into the research of it. And I just became fascinated by everything.
That's great. And so, well, it's not great that this exists, but it's great that you were able to dive into to this passion. Have you always known that you wanted to do this research, or was it like once you decided to do a polygens project, it kind of like formed?
I've always been interested in neuroscience and genetics, but really once I became involved in polygens, I sort of discovered my love for discovering like genetics in terms of neuroscience and mental disorders.
Gotcha.
Yeah.
Would you say you know what you want to do next in this field of research at all?
Honestly, yeah, definitely. I think that it would be so cool to be a part of any sort of research relating to genetics and mental illnesses and mental disorders, because there is so much that we don't know right now. And just being able to answer one of those questions would be amazing.
Yes, I fully agree with that as well. I have a question that came in from one of our audience members. Do you feel that these genetic factors are sensitive to environmental influence? And would that be an important relationship to further study?
Yes, definitely. Environmental influence, it hasn't been researched as much, but there is a definite connection and they are definite factors in this sort of genetics. For example, like if you are in a stressful situation, it has been researched and it has been confirmed that you are more likely to develop schizophrenia in later years. So I think that that is a definite yes.
Gotcha. Yeah, there's, I'm sure, so much left to dig into for this. I don't know if you are, I asked the other two panelists, but are you a senior? Are you a junior in high school?
Yeah, I'm a senior in high school.
Great, awesome. So I'm sure, going into college, there's a lot more that you could do with this. So
yeah, I'm really excited about that.
awesome. So I guess I'll let the other two panelists come back and I'll just ask a few more general questions to you all before we finish up. Are you all still here? Hello.
Hi.
Serena. And Ayush, I know you had maybe issues with your camera earlier.
I'm here, just my camera can't turn on.
Okay, no worries at all. I was just wondering, you guys all have gone through the polygens experience and now you're through it. What would you say is something that you wish you had known before you started the project about research, about conducting research? And is there anything that you've learned about conducting research? And I'll let maybe Serena go first, if that's okay.
Yeah, I would say through conducting research, one thing that I didn't realize is you do a lot of research on a lot of things that are kind of unrelated or maybe a little bit related, and you end up with all this information, but I would say 20% of what I actually researched went into my paper and my project. So it's good and bad. You spend a lot of time, but I know so much about this topic and this field now, even if it wasn't displayed in my project. There's so much that I learned from this just through doing this research project.
Awesome. Yeah, that makes sense. Maya, you want to go?
Oh, yeah, I couldn't agree more. I have an entire bookmarks folder just of every single thing that I found, and I went on many, many deep dives about this. And so you just learn so much and you absorb so much, and you do only use 20% of it in your projects, but now you just have all of this information, and it's really cool actually to think about.
Awesome. Ayush, do you agree? Do you have other thoughts on this?
Yeah, pretty much. I agree with everyone, everything that everyone said. But in my project specifically, I didn't do as much research as other people. I did a lot of coding and testing. And regarding that, I think testing, you don't really realize how long like stuff's going to take. You might just get like an error here and there, but those errors can take like days to fix, and it can get super annoying at times. That's what I realized.
Yes, that definitely, I mean, research is such a long and drawn out time. So I'm sure you've all had your challenges, but glad you pushed through and glad you made it to your end. And thanks for all presenting great presentations. We do have actually one more question that just came in for all of you. So what was your experience of working with a mentor, but actually leading your own learning? I'm sure this doesn't happen often in school. So what was it like kind of, being the champion for yourself in this project?
I thought it was amazing. I had a really great mentor. And first of all, it was just really interesting to hear about what he does, like what his research is and his experiences are. And that was really cool. And obviously, yeah, leading my own project was a little intimidating because I've never done a project like this and I didn't really know where to start, but he like guided me and I was able to put in everything I wanted to put in, and he just helped like clean it up a little bit and make it more professional. And it was really cool.
Awesome. Great to hear. How about Maya or Ayush? Do you have any thoughts on this?
Yeah, I wasn't expecting a collaborative experience when I came into this, but it was really, really great. My mentor was awesome. And we sort of just like worked together, and every time I had an idea, she was like, great, put that down, write that on paper or whatever. And every time she had an idea, I was like, okay, I gotta keep that in mind. And then I wrote it down, and it was sort of just like bouncing off of each other, and I loved it. And I'm glad that I got this experience, because I probably wouldn't have gotten it if I had just stayed in school or only going off of school.
Yeah, that's awesome to hear. I'm really glad that you guys had great experiences with your mentors. So, Ayush, do you have any thoughts?
Yeah, I had a really great experience with my mentor. He was kind of just really pushing me in the right direction the whole time, and that really helped me because this is like the first project I've done like this, so I didn't really know where to start either, and he kind of really guided me, so that was really helpful.
Yeah, was this all of your first like independent research projects, or have you done things in the past?
Yeah, nothing to this scale.
Yeah, this was my first.
Cool. That's really exciting. I'm sure you guys will all continue down some sort of research in the future. So I'm glad that you guys kind of got to experience this firsthand first. So yeah, that is all the questions that have come in. I really appreciate you all for coming and presenting today. I definitely learned some stuff today from you guys. So yeah, I really appreciate it. And thank you so much again for doing a Polygens project, and good luck in college as well, since you all are seniors.
Thank you.
Thank you.
I actually have a question.
Oh, yep, go for it.
So when do we get to know how we did?
That is a great question. I am not 100% sure. I can let you know though. Does anyone in the audience attendee know? If not, then I will get back to you, I'm sure, about when the results are posted. We will let you know over the next week.
Great.
So stay tuned. And you guys all did great. So thank you again. Any other questions? Okay. Well, have a nice Saturday.
Thank you.
Thank you.
Bye. Bye.